Hello and welcome back to my channel. In today's video, let's look at Lac Operon. Lac Operon is an example of how cells can modify their genetic response to the environmental change. In this case, the environmental change being the availability of lactose and glucose. So for bacteria like E. coli, glucose is the preferred source or metabolic substrate for energy. And in the presence of glucose, Lac Operon gene is not activated. In the presence of lactose, the Lac Operon gene is activated. So when you have very high glucose and you have no lactose available, Lac Operon gene is not expressed. When you have low glucose and you have no lactose available, so low glucose and no lactose available, still not no Lac Operon is expressed because you need lactose to be present for the Lac Operon gene to be expressed. So when you have low glucose and you have high lactose, that is when strongly Lac Operon will be expressed. Like here, low glucose and lactose is available. Lac gene are strongly expressed. And when you have high glucose but lactose is still available, that's when very low or basal expression. So high glucose and lactose is available. Lactose is present. That's when you have a basal, very mild expression of Lac Operon gene. So the Lac Operon gene has many segments. So these are the ZYA. And these are your Lac Operon genes. And before the Lac Operon gene, there are many sites which regulates the transcriptions of these genes. So number one thing is a CAMP site. CAMP site is going to increase transcription. And to increase transcription, you need CAP to attach to the CAP site. Now, when glucose is present, glucose inhibits adenylase cyclase. Adenylase cyclase is an enzyme that converts ATP to CAMP. CAMP activates CAP. CAP gets bind to the CAP site and it causes transcription of ZYA Lac Operon genes. When you have glucose, it inhibits adenylase cyclase, no conversion of ATP to CAP. Therefore, CAP is not activated. Therefore, CAP does not bind to the CAP site and transcription of ZYA does not occur. Promoter is the site where transcription occurs if the CAP binds to the promoter site. After the promoter site is the operator site. Operator site also causes transcription of the ZYA Lac Operon gene, but operator is a site where repressor protein is bound. Repressor protein inhibits the transcription of the Lac Operon gene. So in the presence of lactose, you have a allolactose. In the presence of lactose, you have a inducer allolactose that binds to the repressor protein and removes this repressor from the operator site and hence causes increase in the transcription of the Lac Operon gene. So there are two ways. One is when you have high glucose. When you have high glucose, you have inhibition of the adenylase cyclase enzyme. And when you have lactose available, so unbinds repressor protein from repressor oppressor site, increase in the transcription.